Okay, in framework of our session, I would like to talk about the online databases and their potential application in calibration and validation of QSP models. So the three different topics will be discussed in framework of our section. First, importance of publicly available data for QSP modeling. And second and third topic, we would like to discuss the two different databases, CYTOCON DB and 5DB. Second database will be discussed by a separate speaker. So let's start with the introduction. QSP model is a complex mathematical object representing our knowledge and structure and regulations of biological system and PK/PD of a drug in terms of equations and parameter values. Parameters are identified against available in vitro and in vivo data in such a way to constrain values of variables in experimentally measured ranges. So availability of data and ease of their use is of a special importance for QSP model calibration and validation. Here you can see the example of the QSP model. This type of the model typically includes several compartments, and several cells and cell states, as well as the transition between the cell and cell states. This transition is typically regulated by the cytokines. What type of the data do we need to calibrate and validate such type of the models? First type, these are in vivo measured cell and cytokine concentration. Second type is in vitro measured data describing effect of the cytokines on cell dynamics. So the objectives of our session are to overview difficulties encountered on the way of collection, processing, and use of this type of the data, and to describe CYTOCON DB and 5DB as tools allowing to overcome the difficulties and facilitating calibration and validation of QSP models. Let's start with discussion of the first database, CYTOCON DB. But before, I would like to say a couple of words about characteristic feature of baseline in vivo cell and cytokine data available publicly. This type of the data published in different sources, such type as scientific papers, posters. These data are typically published and measured in different units. Even more, cell concentration measured in percentages of larger cell population sometimes. For example, Treg cells count can be measured as percentage of the CD4 T cells. Part of information is quite often lost. For the example above, parent concentration of CD4 T cells may not be published in the source paper. How can we resolve all these issues? We are developing the CYTOCON DB for these purposes. This is manually curated database which contains information about baseline levels of cells and cytokines in vivo concentration, measured in different tissues of healthy subjects and the patients suffering from different diseases. Key features implemented in CYTOCON DB are multiple diseases, including different diseases inside of a family of disease, for example, different cancers, bladder, breast, cervical, colorectal, and so on. Automatic conversion of the source units to unified units, picomole for molecules and kilocell per liter for cells. Recalculation of relative or percentage level of cells in tissues to absolute units, kilocell per liter, and many others. Here you can see example of the usage of the CYTOCON DB. In framework of these efforts, we extracted and compare of different cell counts in their ranges measured in different tissues of healthy subjects, melanoma, and non-small cell lung cancer patients. If we analyzed all these plots, we can come to several conclusions. One of them, that amount of myeloid dendritic cells and natural killer cells lower in melanoma tumor than in non-small cell lung cancer tumor. And final slide represents our CYTOCON DB team, our contact details, and web resources where the database is located. Thank you for attention.